Hello, my name is Hendrik von Buren. I'm the German General Manager, Research and Development of the global biopharmaceutical company AbbVie at the affiliate in Ludwigshafen. I've been with the company since 1999, and it was just 2 years before my entry when my colleagues had a compound in clinical development that would revolutionize the treatment of rheumatoid arthritis, or RA, and other 14 chronic autoimmune diseases over the lapse of 20 years. But let's start with the beginning, the pioneering work done by my research fellows. In the 1980s and early 1990s, rheumatology practices were literally full of patients in wheelchairs with curved limbs and incredible back pain. All that physicians were able to offer at that time were cytostatics and surgery in which damaged joints were replaced. At this time, my colleagues examined the biological functions of a body's own protein, the so-called tumour necrosis factor, or TNF. In acute conditions such as septic shock, the levels of TNF are extremely high because the body releases TNF to fight the infection. However, a surprisingly new finding at that time was that the protein is also present in large amounts in people with RA. This group of people had no infections, so why were their TNF levels so high? This question led researchers to the idea of focusing on this disease and developing a fully human antibody. A biologic that could neutralize the TNF, but that should be at the same time relatively well tolerated. RA is not curable, so the active ingredient had to be suitable for the treatment of a chronic disease. The approval of the drug class of TNF inhibitors, such as D2E7, Humira, was a true paradigm shift in the treatment of RA. These drugs fundamentally change patients' lives because they can keep the disease under control so well that patients are able to return to normal life. The quality of life of those affected has therefore increased enormously. But we did not stop there. In recent years, we have further developed Humira clinically to use it in other therapeutic areas, such as psoriasis, ulcerative colitis and Crohn's disease, as well as in children and adolescents. At the same time, we have continued our research to provide patients with additional treatment options. This is also an urgent necessity for chronic diseases. Recently, we have been able to continue the success story of D2E7 with another therapeutic antibody and a small molecule. This demonstrates the importance of long-term intensive research and development of innovative therapies. It is the only way medical progress can be achieved.